Hi, I'm Saloni, and my research topic was analyzing the relationship between human endogenous retroviruses and multiple sclerosis, essentially HERV prevalence as a factor of MS expression. And I'm going to go more into this now. So, what is a human endogenous retrovirus? So, HERVs, so basically, normal viruses replicate by injecting their DNA into the host cell and then hijacking that host cell's mechanism in order to create RNA and proteins and such in order to create mature viruses, which will then be released from the host cell. Retroviruses do the opposite. So what they do is they actually insert RNA into the host cell. They hijack the machinery in order to create DNA. And then there's one other key thing that they do. They also, their DNA, the DNA of the retrovirus also binds to the DNA of the host cell, essentially creating a new DNA sequence, right? Because you have the host cell's DNA plus the viral DNA. So this process kind of shows what the retrovirus does. You have your retrovirus inserting into the host cell. You're going to have reverse transcription. So you're going to have mRNA to DNA, which is then going to be integrated into the host cell's DNA. Now, where does this fit into the human body? So normally, right, if an organism is infected with a virus, this isn't going to affect the offspring. So if a person is infected with a virus, considering current times, we can say maybe coronavirus, and then, 10 years later or so, they have offspring. That viral DNA isn't going to be passed on to their offspring. However, in certain cases, if the virus infects a germline cell, meaning a sperm or egg cell, then potentially viral DNA could be passed down onto the offspring. And so that has actually happened multiple times throughout human history, right? So we have, say, maybe some of our early ancestors, pre-human, pre-Homo sapien ancestors being infected with these retroviruses and then passing them down to their offspring and actually aiding in the process of revolution. Not revolution, evolution. And so what does the term human endogenous retrovirus mean? So human, self-explanatory. Endogenous means within, and then retrovirus. So actually about 8% of our DNA is actually made up of these HERVs, human endogenous retroviruses. Okay, so that was a little bit of background on what HERVs are. Now we're going to move on to MS, so multiple sclerosis. So multiple sclerosis is essentially an autoimmune neurological disease. What happens is that the body starts attacking itself. And so basically within the neurons of the brain, there is this portion of the neuron called the axon. The axon facilitates communication with other neurons, other cells. And so there are these long thread-like fibers, and they have these things called myelin sheaths wrapped around them. The function of myelin sheaths is kind of like insulation, right? So neurons pass electrical signals to each other, and axons do a similar thing, right? If you think of a wire, passing electrical signals along, you might think of a copper surrounding the wire in order to help facilitate the transmission of the electrical signals or the electricity. So we have the same kind of concept here. Myelin sheaths are made up of fats that surround the axons in order to help facilitate electrical transmissions from cell to cell. And you can kind of see that here in this picture on the right. I'm not sure if you're able to see my cursor, but on the picture on the right, we see two axons. We see a healthy axon with those tan colored boxes all along the axon. That's what and obviously this is an artist's rendition, but this is what a normal axon might look like. The one on the right, however, is a demyelinated axon, meaning the myelin sheaths surrounding the axons have become degraded. And so we see that here. Another thing that occurs with multiple sclerosis or MS is that the brain and the spinal cord start experiencing lesions or scarring. And so we see that on with the picture on the left. Oh, didn't mean to change my slide. In the picture on the left, firstly we see a normal brain, but then in the brain scan next to it, we see a MS affected brain. And the white areas specifically show areas of lesions or scarring. And so basically, multiple sclerosis has many symptoms, namely because multiple sclerosis affects cell communication and more specifically neuronal communication. Um, this can lead to a whole host of symptoms. It can cause symptoms such as fatigue, difficulty walking, as this involves, coordination of muscles, tremors as well, vision problems, and like I said earlier, it causes lesions internally, which means damage or scarring within the brain and spinal cord. And this is because of inflammation caused by the body attacking the myelin sheaths. Okay, so now that we've concluded the background, again, we're going to revisit my question, which is MS partially or completely caused by the body exhibiting an autoimmune response to the presence of HERVs in the body? So let me expand upon that a bit further. So an autoimmune response basically means it's an over-exaggerated response of the body to what it believes are foreign invaders. As you might remember from what I said earlier, HERVs are in our human genome, right? But originally they weren't there. They're part of a different, I'm not sure if you can call viruses organisms since they don't have the ability to replicate themselves, but they come from a foreign molecule or foreign object. So essentially, it's been theorized that multiple sclerosis in particular, along with possibly other neurodegenerative diseases, which are usually a result of an autoimmune reaction, are caused by the body attacking what it thinks is a foreign invader, when in reality, it's just a part of the human genome. But because it has an exogenous root, it thinks, or the body's immune system reacts in that way, attacking itself because it believes that these are foreign invaders rather than a part of the body itself. So now we're going to be going into some of the a bit further into detail about viruses and retroviruses in general. So we're going to be going over two specific terms, GAG proteins and ENV proteins, because they're going to be crucial to the research that I talk about later. So firstly, let's start off with GAG proteins. It's a acronym for matrix and capsid group antigens, so GAG, GAG. Basically, these proteins are within a virus, and these antigens are proteins within a virus, and they're targeted by this enzyme called viral protease. Protease is an enzyme that cuts proteins, and then eventually these segments are reassembled to make the mature virus. So basically, if there are active GAG proteins within a system, a mature virus is being formed. And so I have a little picture here. Oh, didn't mean to skip slides. I have a little picture here. It isn't super important to know about all the functions in this picture, but I did kind of want to point out where the GAG polyprotein is found. So you have your viral RNA in the center, and then as it is a matrix and capsid protein, right, you're going to find it on the outer, somewhere near the outside layers of the virus. And so we see here, I'm not sure if you can see where the arrow leads, but it leads to the brown segments near the outer membrane, or this membrane right here. Basically, their GAG proteins are essential to the formation of mature viruses. So now let's move on to ENV proteins. So ENV proteins are short for envelope proteins. So they code for the viral envelope. So aka membrane, right? So this membrane or this envelope allows viruses to target specific types of cells and then pass their viral RNA through the membrane of the host cells. And ENV proteins, along with GAG proteins, exist in trace amounts in normal brains, but they are not activated normally because the HERVs do not fully form, right? When we, in some time in our genetic history, when we adopted HERVs into our genome, these viruses don't fully form within our bodies. Over time, through evolution, we've kind of changed some of the viral aspects so that it doesn't code for the full virus anymore, or I suppose we have not changed, but due to environmental factors and as a result of evolution, the viral components within our genome have been changed, and so therefore full mature viruses are not formed. So if we take a look at this picture, so HEIV, human immunodeficiency virus, is a virus that is in retrovirus actually. So this diagram is just a quick demonstration of the ENV and GAG proteins I was talking about. Because they're involved with the membranes, you'll find the ENV proteins near the outer layers of the outer segments of the virus. Because that is where the envelope will attach to the envelope of the host cell. Then you find your GAG proteins for example, GAG P17, right beneath the outer layer of the virus and within this inner layer as well, within this inner membrane as well, which houses the RNA. Essentially, both are extremely crucial to the development of the virus. Okay, so now we're going to move on to the studies and the research that I've collected. So this is one specific study conducted by Perron et al. That's just going to be how I refer to this study, as the title is quite long, and this encapsulates the people that have worked on it. So basically, this study analyzed the relationship between the GAG proteins from the HERVW family. So within our body, we have multiple families of retroviruses. Specifically, the W family has been associated with multiple sclerosis and the expression of multiple sclerosis. So essentially, they studied the HERVW GAG proteins. Quite a long name, but that is what the people in this study researched. So basically they found that there is a minimal expression of these GAG proteins within a normal brain, which can be expected. But in the diseased MS brain, there's a large accumulation of HERVW GAG antigens within the axonal part of the demyelinated matter. As you might remember from what I said earlier, MS is caused by demyelination, meaning the breakdown of the myelin sheets in the axons. And so that relates to this perfectly, because the GAG proteins were found in large amounts within the demyelinated portions of the axons. So, and then they also recorded that there seemed to be a relationship between the increased prevalence of functioning GAG antigens and then people that were experiencing greater and greater demyelination. How did they measure this? So what they did is that they used antibodies to counter the GAG antigens, and then this showed up visually on their slides. So as you can see here, we have four diagrams here. Specifically, we're going to be looking at diagrams A and B. So diagram A is a slide which shows a cortical unaffected neuron far from any lesions or demyelinating sites. So this is within a MS-affected brain, but it's far from any demyelination and lesion type sites. And then so in picture A, we don't see anything out of the ordinary, nothing super unusual, But then if we look at picture B, we see an axon in the center of an actively demyelinating lesion. So here we can see that there are a lot of blue circles, and those blue circles are the GAG antigens that we were talking about. Those blue circles represent the GAG antigens that we were talking about, and they show, and we see that there are none in the unaffected neuron. So I just wanted to show that picture real quick because this was from the research study conducted by Perron et al. And I thought it was interesting to show visually what specifically the researchers were looking for. Now we're going to move on to a study done by Laska et al. So basically this study, this graph that you see here shows the amount of HERVW GAG antigens in plasma, which means blood, across trial groups. Basically, so as we said earlier, the GAG proteins exist within the brain in small amounts because they are remnants of the HERVs they originate from, right? However, they normally remain stagnant and unactivated. Additionally, you can see here that the healthy controls and, interestingly, the patients with non-active MS have relatively low rates of GAG proteins, but then patients with active MS have 3 times the amount of GAG proteins. So this brings in another interesting topic, or another interesting point of thought, that people with non-active MS do not seem to have large amounts of GAG proteins. It's only people that have active demyelination occurring rather than stable. Stable just means that there's no demyelination. Demyelination has happened at some point, but at this point, there is no further demyelination happening. Patients with active MS, on the other hand, have increasing rates or increasing amounts of demyelination happening. And so we see in this graph that the GAG proteins in healthy people and healthy brains and in non-active MS brains have low rates of GAG proteins. But then people with active MS, meaning lots of demyelination happening, have high rates of GAG proteins. So clearly there has to be some sort of relationship between demyelination and the prevalence of GAG proteins. So now we're going to move on to this study conducted by Mameli et al. I hope I'm not butchering this name. So it's a similar study, but instead of the previous ones, this one focuses on ENV proteins in patients. So in patients with MS, there was about 20 times the amount of ENV proteins compared to the contrasting groups. So in this graph, you can see that there are 3 groups. So NBC stands for normal brain controls, so that's your control group. OND stands for other neurological diseases, and MS obviously stands for multiple sclerosis. So clearly there is a clear relationship between the prevalence of multiple sclerosis and the prevalence of ENV proteins. Moving on to Brudek et al. So with Brudek et al., excuse me, this isn't loading. Okay, so they found that patients with active MS had significantly higher rates of the ENV proteins found on the surface of B cells and monocytes. So what does that mean? So B cells are cells that fight infectious bacteria and viruses, and they do so by releasing antibodies, which then bind to the invading cell, and mark it for destruction. And then monocytes actually do the destruction of the invading bacterium or virus. B cells just do the marking. So basically what Brudek et al. Did is that they measured the amount of HERV proteins by inserting antibodies and then studying the immunofluorescent activity. And what they found, so here in this graph that you see on the right, you see a line for CD19+, which is the code that they used for B cells. And then you also see a dashed line for monocytes. So in the healthy control, so HC, and then stable SMS patients, as the amount of antibodies they injected increased, the amount of immunofluorescent activity, meaning, and so that's basically how many antigens there were. So in this study, they did ENV antigens. It stayed stable, right? So this means is if this means that even though the amount of antibodies they were releasing was increasing, the amount of ENV activity was not. So this means that there's a relatively small amount of ENV proteins, right? Because if I have a little bit of ENV proteins, right, and then I keep releasing antibodies, right, it's just going to flare up a little, right? And no matter how much and how many antibodies you insert into the system, there's just going to be a small weak response. However, if I have a lot of antibodies, if I release a little bit of antibodies, you're going to have some activity, and as the amount of antibodies you're releasing into the system increases, the amount of ENV activity will also increase. And that's what we see here, right? So in the active MS patients, we see that as the amount of antibodies, which is indicated by the x-axis, increases, the amount of ENV activity is also increasing. So basically, from my explanation with my hands and this graph, we can conclude that patients with healthy brains and stable MS patients tend to have lower amounts of ENV activity. And then patients with active MS tend to have high rates of ENV activity. Okay, moving on to Aru et al. So essentially, it's pretty clear based on all the studies that I've shown you and along with others, but for the sake of time and for the sake of being concise, I didn't include all of them in here. You can see that there's a clear connection between ENV and GAG proteins, which are related to HERVs, and the prevalence and expression of multiple sclerosis. Again, I say prevalence and expression because patients with MS, obviously it's prevalent within their bodies, but it's not being expressed actively, right? So if I have stable MS, it's within my body, right? But because the demyelination isn't progressing, the expression is not as much as a patient with active MS, right? So as I was doing further studies, right, I realized that perhaps HERVs are not the only reason for MS. Again, I didn't go into that, but it was an interesting point to be brought up. So in a study conducted by Aru et al., we see that there's a high rate of MS in Sardinia. So that's this little, in this picture, it's colored black. And it's a little island off the coast of Italy, and it has a high rate of MS compared to other European regions. Additionally, across the world, we see that Africa as a continent and ethnically African people tend to have lower rates of MS compared to Asia, and then people of European descent tend to have the highest rates of MS population-wise. So this brings up an interesting point because if the only factor for multiple sclerosis was HERVs, which have been a part of the human genome for millions of years, right? They shouldn't fluctuate within regions, right? Because people of European descent and people of Asian descent, for example, or different varying populations across varying ethnic descents across the globe, they're still Homo sapiens, right? And so all these genetic differences arose after the Homo sapiens species was fully established. But since HERVs are intrinsic to humans and to Homo sapiens as a whole, you wouldn't expect to see differences across populations. And so this brings me to my conclusion that ultimately there seems to be a direct relationship between an autoimmune response of the body to human endogenous retroviruses and the occurrence of multiple sclerosis. The prevalence of retrovirus-related proteins, so specifically GAG and ENV proteins, as we as we studied through these research papers and these clinical trials, that these retrovirus-related proteins lead to the expression of MS. However, there are other outside factors influencing the development of MS, which need to be studied in greater detail. However, this does not deny or degrade the conclusion that HERVs are directly responsible for the prevalence and occurrence of MS. So that concludes all my research. Before I finish, I do want to go over why and how I was intrigued by this topic. So when I was first assigned this research topic, my teacher, Ms. Baker, said that we could do anything we wanted. And so that's daunting, right? As a student, when you're told you can do anything you want, when you're so used to being told by the teachers and superiors that this is exactly what you need to do, it's extremely daunting. So I was at my wit's end, and I was trying to find topics and hitting dead ends and not being able to find anything original. And so ultimately I gave up and I started reading. And so I was reading a sci-fi novel at the time, and the author isn't super educated in these topics, but he was talking about HERVs as a justification for whichever scientific concept, sci-fi type concept he was trying to bring up. And so I was like, what are HERVs? Because I was like, I wasn't sure if they were a real thing or if they were something that this author just made up for his own intents and purposes, for the plot as a whole. And so I decided to do some research, and I found out that yes, HERVs are real. So I kind of went down this rabbit hole of trying to understand what they are. So firstly, I tried to understand the mechanisms of how viruses work, because I didn't actually know how viruses work. So I watched 3 or 4 YouTube videos on how viruses work, and then I watched 3 or 4 YouTube videos on how retroviruses work. And then I found some TED Talks and more YouTube videos on what HERVs are, so the human aspect to the retroviruses, and how they actually contributed to our evolution, and how they're an intrinsic part of our DNA now, how they make up about 8% of our DNA. And so once I was satisfied with all this, I was like, oh, okay, great. And then there was a video playing in the background while I was doing homework for another class. And then I heard the professor or the researcher that was talking about HERVs bring up an interesting thing. He said that there seems to be, based on clinical trials and studies, a relationship between HERVs and multiple sclerosis. And so I was like, that's interesting, because I knew about multiple sclerosis because I'd learned about it before. So I went back and I watched the video in detail, and then that's when it hit. I knew I wanted to do this topic, essentially because I had given up and I read a book. So to the maker of that book, the book was called Atlantis Gene. I'm not sure who the author is, but I salute you. Thank you for unintentionally spurring me on this journey, and thank you so much for watching through this video and listening to my topic about HERVs and multiple sclerosis. Thank you so much, and have a great day. Bye.